Let's kind of refresh our memory a little bit. Which gland initiates the entire reproductive cycle? What starts everything?
Hypothalamus.
Very good. Hypothalamus is starting everything. It is secreting a hormone which is known as?
The GnRH.
Very good. Gonadotropin releasing hormone. GnRH is working on?
Anterior pituitary gland.
Very good. And as a result, this gland is going to be secreting two hormones known as the FSH and LH, as you remember, right? What is the purpose of FSH?
It causes
Like
the
the follicle.
Stimulate the follicle.
Very good, very good. It chooses the follicles, it makes them grow, it's stimulating them to enlarge, and etc. According to anatomy department, in 2 ovaries, we have, starting from the puberty,
15 to 20.
starting from the puberty, total amount of follicles in 2 ovaries are?
40,000.
40,000, very good. We have 20K in one and 20K in another, right? So when you're starting the puberty, as you remember, this number is very important. But don't forget the real number is more like 240,000, right? Every month out of these 40,000, we choose how many?
15 to 20.
15 to 20. And how many of them will mature? How many of them will ovulate? Only one. But don't forget, it can be 2. How do we call the follicles that we have made during embryological development?
Primordial.
Primordial. What does a primordial follicle consist of?
Squamous epithelium.
So first of all, inside of it, you will have the primary oocyte, and this primary oocyte is surrounded by the flat follicular cells, right? So squamous follicular cells. How do you know that only 15 to 20 of the follicles are the chosen ones?
They will reach out to the FSH. They will raise a hand.
So every month, randomly, 15 to 20, right, out of the 40,000, they will have some kind of receptor, and this receptor is for the FSH. So randomly, they express the receptor for the FSH. So FSH comes in, binds to this receptor, and forces these follicles to grow. We said primordial follicle becomes primary. You remember this is a mistake in the book, right? So this is going to be the first stage of the primary. This is the second stage of the primary. How do we call these cells? These cells, what is their name?
Granulosa.
Granulosa cells. Granulosa cells are basically stratified cuboidal follicular cells, as you remember, right? So these cells are stratified cuboidal follicular cells. What do?
Secrete the zona pellucida.
They secrete zona pellucida, yeah, and the zona pellucida is going to be coating the primary oocyte inside. Zona pellucida, don't forget, is a glycoprotein, right? So this is a glycoprotein right there. How do we call the connective tissue of the ovary that surrounds each follicle?
Theca follicula.
Very good. We're going to call it theca folliculi. So theca folliculi, this is just the connective tissue of the ovary, which directly surrounds the follicles. The next stage after the primary follicle is the secondary follicle. And as you remember, secondary follicle has a little tiny cavity. And again, I forgot the picture. Okay, I will put it in the group. After this class, I will put it in the group for sure. Okay, so secondary follicle will have just a little tiny space right here that is surrounded by the granulosa cells. Last class, I told you that 15 to 20 of the follicles, right? So they're the chosen ones. 15 to 20 can reach the secondary follicle stage, but only one of them is going to reach the final stage, which is the tertiary follicle. So let's take a look. What do we see in the tertiary follicle? First thing first, this is the area where theca folliculi used to be. I repeat, theca folliculi, this is the connective tissue, which is outside of the follicle. This is connective tissue of the ovary, which is outside of the follicle. Theca folliculi in the tertiary follicle differentiates into two layers. We have the theca interna and theca externa. In the book, they're the opposite. So it's another mistake in the book. In the book, theca interna is outside, theca externa is inside. But use your logic, right? Theca interna should be the inner one. Theca externa should be the outer one. Theca externa consists of collagen fibers. Okay, so it simply consists of collagen fibers and some kind of interstitial cells, but they aren't important for you this semester. Next semester, we'll talk more details. So theca externa, just collagen fibers. Theca interna has capillaries. This is the vascular part of the follicle, and it also possesses theca interstitial cells. These theca interstitial cells now resemble fibroblasts. They look like little tiny fibroblasts for now. So only in the tertiary follicle stage, theca folliculi differentiates into two, right? So into the theca interna and theca externa. Another thing that you notice that the antrum, or the space of the tertiary follicle, is definitely becoming much larger, right? As you remember, in the secondary follicle, we had a very little space. It was a very, very tiny space right here. But in the tertiary follicle, you can see that it is a huge space, huge antrum. This antrum is filled with a fluid, which is called follicular fluid, but Professor Rosa calls it liquor. Liquor is a name for any fluid that has lots of proteins in it. So protein-rich fluid is actually should be called liquor. So inside of this tertiary follicle, we have a bunch of liquor. Which cells are outside of this antrum? Well, these are stratified cuboidal follicular cells, which we call granulosa cells. But granulosa cells keep on proliferating and surrounding your primary oocyte. So inside, you're actually going to see the primary oocyte. The granulosa cells that surround your primary oocyte make a structure which is called cumulus oophorus. So cumulus oophorus is not just this area, it's all of it according to our university. All of this area is cumulus oophorus. It's just granulosa cells which are within the proximity of the oocyte. Okay, so it's just granulosa cells which are very much nearby of your oocyte. Corona radiata is a very special structure that appears after ovulation. Ovulation means that this tertiary follicle has to rupture. It's like a zit that is just rupturing. And oocyte is releasing, it's flying towards the fallopian tube. That's ovulation, when this follicle ruptures, right? The granulosa cells, which go with the oocyte during ovulation, are called corona radiata. So in a way, corona radiata is the remnant of the cumulus oophorus, right? Cumulus oophorus is all of these granulosa cells, but after ovulation, only the granulosa cells that are touching the oocytes are going to go with it to the fallopian tube. So that's what we call corona radiata. So is the structure of the tertiary follicle clear? There is something which is also called Graafian follicle. Graafian follicle is tertiary follicle 24 hours before ovulation. So 24 hours before ovulation, this tertiary follicle will be swelling up. It increases to 25 millimeters in diameter. It becomes super huge. It's ready to burst, right? I repeat, when this follicle ruptures, this is called ovulation. So Graafian follicle is exactly 24 hours before ovulation. It is tertiary, it's just super, super big. Two things you need to remember. we have two hormones. We have FSH and we have LH, right? We have just talked about them. We said that FSH has a job of stimulating your follicle to grow, to reach this final stage, this tertiary stage. But you're going to see that it has additional function. FSH binds to the theca interna cells, and theca interna cells secrete something which is like a precursor of the estrogen. So when FSH binds to the theca interna cells make precursor of the estrogen. This precursor is taken up by the granulosa cells and converted to the active form, which is estrogen itself. So is this clear? So who makes the estrogen? You can't really answer that because technically theca interna gives you the precursor that is needed, right? But granulosa cells are actually converting that precursor to become the estrogen itself. This is the function of FSH. It is done by the FSH stimulation. But what does LH do? What is the function of LH in the follicle? LH will be binding to the granulosa cells and they will secrete follicular fluid. So the reason that this antrum becomes bigger, because there is more fluid. Who tells you to secrete more fluid there? Well, of course, your LH. Is it clear? Okay, we talked about the diplotin stage. Could you remind me what is that?
It's the stage where the egg is frozen.
Yeah, but say a bit more. So primary oocyte enters what?
The prophase stage of
Meiosis 1. So it is on the way of becoming secondary oocyte, right? But we arrest it in the prophase. Who arrests it in the prophase?
Omi?
Oh my.
But
Omai.
who makes the omai? Who secretes omai?
Follicular cell.
Follicular cell, yeah. So follicular cell is the one who is secreting omai, and as a result, it is arresting it in the diplotin stage. Okay, everything I'm talking about here, I want to do it on the graph. This graph, guys, it's crucial. Next semester, you learn it in both, in anatomy and in physio. This semester, so many questions are going to come from here. We have to look at the ovarian cycle. We have to see what is happening with the hormones, right? And how are the changes are, right? So due to the hormones. Who knows, how long is the ovarian cycle in females?
30 days.
28 days on average, 28. Okay, that's how long the ovarian cycle is. In some it is 27, in some it is 31. So we're gonna draw a similar graph here. This is going to be the concentration of the hormone, 100% being the highest, right? And this is going to be time. So this is day 1. This is day 28. This is how long the ovarian cycle is. What happens on day 1? How do you start counting that this is day 1?
Your body is rich with FSH and LH.
How does a female know that this is the first day of her ovarian cycle?
After menstruation.
Not after, the day she gets menstruation. The first day that she got her menstruation, this is the first day of ovarian cycle, always. So first day is always the first day of menstruation. On average, menstruation is about 5 days, right? So menstruation finishes usually 5 days. What happens on day 14? What happens on middle of the cycle?
ovulation.
Ovulation. Day 14 is the time when typically ovulation happens. Ovulation, we have said, is basically when the follicle ruptures, right? So your Graafian or tertiary follicle will be rupturing and releasing your oocyte. You can see that day 14 is kind of in the middle of your ovarian cycle. This whole thing is the whole ovarian cycle, but day 14 is in the middle. As a result, it sort of divides ovarian cycle into two. The first phase is known as the follicular phase of the ovarian cycle. So from day 1 to day 14. Guess what we're doing during follicular phase? What are we doing?
Maturing the follicle.
We're making the follicles. We're converting primordial to become primary, to become secondary, to become tertiary, until one of them reaches this mature stage, right? So in the follicular phase, we're simply taking care of the follicles. We're stimulating them to grow. So what do you think? Which hormone should be the highest in this phase, in the follicular phase? FSH or LH?
FSH.
FSH, of course. In this graph, it's shown very weirdly. FSH is actually rising. It keeps on rising, and in a certain time, it reaches its highest concentration. And then you're going to see that it will drop a little bit. Okay, it will drop a little bit, and then it will rise just a bit more around day 14. I will explain why. And then it will be dropping dramatically and just going down like this, like a normal concentration. So this is FSH concentration right here. In the follicular phase, you can see that it is rising, then dropping just a bit, and then rising again. The second phase is known as the luteal phase. This is where the luteal phase is. And luteal phase is very important because ovulated follicle, right, a ruptured follicle, will undergo some changes. The follicle that remains in the ovary after you ovulated is known as the corpus luteum. As a result, this phase is known as the luteal phase. What do you think? Which hormone will be the highest in the luteal phase?
The LH.
The LH, luteinizing hormone, right? So you can imagine that in the luteal phase of the ovarian cycle, to influence corpus luteum, you will need a luteinizing hormone. So let's talk about how exactly do I ovulate? What is happening exactly? First of all, you remember that we have hypothalamus. You remember that it makes GnRH. My question is, does hypothalamus make GnRH continuously or in the cyclic manner?
Cyclic manner.
In the cyclic manner if you are an adult. If you're an embryo, it makes it continuously. But what are we talking about right now? We're talking about puberty, so you're adult already. So GnRH will be secreted in cycles. When is that cycle? First cycle, first dose of GnRH is approximately 1 day before menstruation. Okay, 1 day before menstruation, we have GnRH hitting very high. The second dose of GnRH is 1 day before ovulation. That's the 2 times when GnRH is high. If GnRH is high, which other hormones will be high as well?
FSH.
FSH and
LH.
LH. So you're expecting spikes in the FSH and in LH at the same time. You're expecting FSH. Let me put it S here, you know this is FSH, right? And L, this is LH. So at the same time as GnRH is spiking, you're expecting that FSH and LH will also spike. We see it here. Do you see? Here we have a spike in FSH. You're expecting that there will be a spike in LH as well. It's also rising. Why? Because that's where the second dose of the GnRH comes in. Is this clear? GnRH, just twice it pushes. If it pushes, you have spikes in the FSH and LH. In the follicular phase, we don't really need LH, because what is the function of LH? What does it do to my follicle? I just told you. What does it do? What does LH do?
It increase the fluid inside.
Very good. It increases the fluid inside of the follicle. It binds to granulosa cells and causes granulosa cells to secrete follicular fluid into the antrum. So as a result, you're going to see that the amount of LH in the follicular phase, in the first phase, is very low. It's not really doing much because we don't need the fluid yet. We're just growing the follicles. The dominant hormone is definitely FSH, right? You can see that FSH is definitely rising. But I told you, what is another function of FSH? Another function of FSH is to bind to fica interna. So fica interna can produce estrogen precursor, right? Who will take up this estrogen precursor?
Granulosa cells.
Granulosa cells, and these granulosa cells are going to convert estrogen precursor to estrogen. So if FSH is rising, which other hormone should be rising as well?
Estrogen.
Estrogen. As you can see, estrogen is spiking during the follicular phase as well. The more follicles you make, the more granulosa cells there are, the more FSH there is to stimulate these follicles to grow, the more FSH is asking your granulosa cells to secrete estrogen. So which hormone is dominant during the follicular phase? Estrogen. Which gonadotropin is dominant during the follicular phase? FSH. FSH is a gonadotropin, right? But pay
Ask a
attention.
question.
Yeah.
Isn't the estrogen is going to be in the lumen of the follicle?
But don't forget, we actually have blood vessels. I go back. These
Oh,
are
right. Yeah.
internal blood vessels. A little bit will go to the blood. Okay, so you're right to say it will be, lots of it will be in the lumen. You're right, but also will be in the blood. We have blood vessels right here, so I will send it to the circulation for sure. Okay, so we have said that this is the estrogen right here. The big question that you should ask yourself is why ovulation even happens. Why does this follicle rupture? What could cause this follicle to rupture? What do you think?
The increase in size.
Exactly. And how can you increase in size?
LH.
Water that inside.
Very good. LH. And what is LH doing to your granulosa cells? It asks them to add
Fluid.
fluid. So the reason that you ovulate is because of this. This has a name. This is known as the LH surge. LH surge is a sudden rise in the LH concentration. Do you see? Suddenly, LH is rising up. If I have too much of LH, as a result, I'm going to have too much follicular fluid in the follicle. It cannot withhold that much pressure, so it bursts. This is called ovulation. The big question is, what causes LH surge? Why LH surge is happening? Next year in physiology, you're going to learn this amazing analogy. I love this analogy. If we imagine that LH is fire, estrogen is gasoline. So what's going to happen if you put just a bit of gasoline into the fire? What's going to happen?
More fire.
Okay, so it's going to literally blow up. So estrogen has to pass a certain threshold to cause a reaction. Chemistry 101. Okay, to cause a reaction, you need to pass a threshold. So when estrogen is rising, eventually it passes its normal concentration that creates a response in LH. Okay, so that causes LH surge. If I have LH surge, I'm going to have ovulation. So do you get it or no? So what is ovulation?
Rise in LH that cause too much.
What is ovulation itself? What does it mean, ovulation?
Rupture of the
The
follicle.
rupture of
Rupture
the
of the follicle. Which follicle specifically?
The tertiary.
The tertiary or Graafian? Because you remember, one day before,
Yeah.
we're going to call it Graafian, right? So tertiary or Graafian follicle ruptures. What causes the rupture of the follicle?
Hormone.
Men's fluid inside, stimulated by LH.
Which hormone
and,
specifically? Which hormone causes rupture of the follicle?
LH
LH.
LH. And is this
triggered
LH
by
normal?
estrogen.
Is this LH normal concentration or no?
No.
No,
You have to say LH surge. What is responsible for ovulation is actually LH surge, sudden rise in LH. Okay, Ido have said that what causes LH surge?
Estrogen.
Estrogen. Remind me why estrogen is rising.
because of FSH.
Because of FSH. And I really like this question in the past papers. FSH indirectly causes LH surge, correct?
Yes.
Yes.
For sure. Because via the estrogen, it does cause LH surge, for sure. That's what FSH does. But who started this whole thing? Who started the FSH and all of this stuff?
GnRH.
GnRH, hypothalamus, twice have gave you the boost in 2 hormones to make sure that definitely LH surge happens. Okay, let's take a look. I go back to this picture right here. No, I'm going to go here. Okay, so I can draw. This is a tertiary follicle. This tertiary follicle is about to rupture. We have said that this is theca externa. This is theca interna. This is granulosa cells. How do we call this whole thing?
Cumulus ophorus.
Oophorus, very good. Okay, so this structure is known as the cumulus oophorus. What is important is the following. In the Graafian follicle, according to our book, we already have secondary oocyte. 24 hours before ovulation, when LH surge is rising, when it is happening, exactly at that time, your primary oocyte differentiates to become secondary oocyte. So in the Graafian follicle, you have secondary oocyte. In the tertiary follicle, which could be like 2 days before ovulation, you still have primary oocyte. Does it make sense? So 24 hours before you're ovulating, you already have secondary oocyte. So here I have my secondary oocyte surrounded by zona pellucida and cumulus oophorus. LH surge causes the following things. So sudden increase in the LH concentration causes activation of collagenase. In molecular biology, you should have learned that collagenase is the enzyme which is degrading collagen. Where do we have lots of collagen?
The zona pellucida.
Zona pellucida is a
Theca
glycoside.
externa.
Theca externa, very good. In the theca externa, I have only collagen fibers. So LH surge, when you're increasing the LH, right, it causes degradation of theca externa. And think about it, it's good. Collagen is a very strong fiber. We do not want it to surround the follicle. It's much easier to rupture when you don't have collagen surrounding you. So one of the first steps during the LH surge is that it activates collagenase. Collagenase degrades theca externa. So during ovulation, you don't have theca externa. It's not there anymore. The second step is that LH causes your granulosa cells to secrete lots and lots of liquor, lots and lots of follicular fluid is being added into the antrum. That increases the pressure inside. The third step is that prostaglandins are starting to increase due to LH surge. Prostaglandin concentration in the connective tissue of the ovary increases. We already know. What does prostaglandin do?
Causes muscle contraction in the female.
Smooth muscle contraction, right? So it causes smooth muscle contraction. In the connective tissue of the ovary, I have smooth muscle cells. I have one smooth muscle cell here. I have one smooth muscle cell here, one here. If prostaglandins are rising, that means that these smooth muscle cells are starting to contract. Now imagine if they're contracting, what are they doing? They're exerting external pressure onto your follicle. They're literally squeezing your follicle like that. Now, the pressure inside the follicle is high because of the follicular fluid. The pressure outside is also high because the smooth muscles in the connective tissue of the ovary are contracting. So what's eventually going to happen? Eventually, you're going to see that this follicle bursts. It just ruptures. It ruptures. As it ruptures, what's going to happen? You're going to see that your secondary oocyte, surrounded by zona pellucida, surrounded by corona radiata, now flies towards the fallopian tube. I'm not sure if you know, but we need this burst. We need this high pressure because ovary is a bit far away from the fallopian tube. And we literally need to shoot a bullet at the fallopian tube. And that bullet is your oocyte. It's not enough that it ruptures a little bit. It has to rupture a lot. It has to rupture with force to make sure that oocyte can reach fallopian tube because they're far away from each other. This is the ovary, this is the fallopian tube. They're a bit far away from each other, okay? So what is left behind? Could you tell me? Ovulation has happened. Secondary oocyte is in the fallopian tube. That's it, it's there. What is left behind? Do we have theca externa?
No.
No. Do we have theca interna?
No.
Yes, we do. Nothing has happened to it yet. Okay, just a little bit of the follicle has ruptured, but we still have a bit of the theca interna, and we also have
Granulosa cells.
granulosa cells. So what is left after ovulation of the tertiary follicle is just theca interna and granulosa cells. How is the LH after ovulation? Is it high or is it low?
Low.
LH surge. Is it going to be immediately low as on this picture? No, it's going to gradually come down to its normal concentration, LH. It usually takes 9 days for the LH to come down to actually reach its normal concentration. So I repeat the question, right after ovulation, how's the LH? Is it high or is it low?
High.
It's super high. And because it is super high, it causes a process which is known as luteinization. Luteinization basically means that you're force feeding the cells LH and asking them to secrete something in return. Because LH is so high, your theca interna and your granulosa cells are starting to take up this LH, and as a result, secrete 2 hormones. Which 2 hormones are they starting to secrete? Estrogen, and now, most importantly, progesterone. These cells secrete more progesterone. 75% of their secretion is progesterone. So if I go forward, you can see that which hormone starts to rise in the luteal phase. Which hormone is rising?
Progesterone.
Estrogen and progesterone.
So who makes this progesterone?
The corpus. Something.
Corpus
Trachea interna.
corpus luteum, the luteinizing cells, okay? If I may show it to you here again, this is our rupturing follicle. This is our oocyte that is flying towards fallopian tube. What is left behind? Do we have theca externa? No, we only have theca interna and granulosa cells. The LH is high. It's so high that we need to use up this LH. We call this process as luteinization process, right? Luteinizing these cells. We force feed these cells LH, and in return, theca interna and granulosa cells secrete two hormones. They make estrogen and progesterone. Which one do they make more? They make more of progesterone. Agree with me, they're new cells. They're now using LH to secrete new hormones. So as a result, their names change. Now you're going to call them lutein cells. You're going to call them theca interna lutein and granulosa lutein cells. How do we call this structure that consists of lutein cells? We call it corpus luteum. So corpus luteum is the follicle after ovulation, when the follicle has ruptured, right? That's the follicle that is left behind. What does corpus luteum consist of? It consists of the theca interna lutein cell and granulosa lutein cell. What do these two cells do? They secrete estrogen and progesterone. Which one do they secrete more? Progesterone. Is it clear? Now, what is the function of estrogen in uterus? What does estrogen do to uterus? I'm not sure if you know, but the inner wall of the uterus is called endometrium. The inner lining of the uterus is called endometrium. This is the tissue where embryo is going to implant into, right? And the function of the estrogen is to proliferate endometrium. It forces endometrium of the uterus to grow because this hormone is preparing for the embryo. Do you see? We're growing the follicles. We're preparing for the embryo. We're hoping that after ovulation, there's going to be spermatozoa so we can fertilize it, right? But what is the function of progesterone? Progesterone means progestation. It is pro-embryo. It is in the best interest of the embryo to stay in the endometrium. But how can it stay in the endometrium? Only if endometrium persists, only if it stays thick. To simplify it for you, estrogen makes endometrium thick. Progesterone keeps it thick. It maintains its thickness. If I don't have an embryo, I didn't have sex, I don't have an embryo, what's going to happen to LH? Is it going to be still high? I don't have an embryo, nothing is happening. Eventually, LH
will drop down.
it goes down. Okay, how long? 9 days it usually takes, right? So for the LH to come down. Who needs LH to function?
The corpus luteum.
Corpus luteum. What happens if there is no LH? The corpus luteum will die. Dead corpus luteum is called corpus albicans. It's white. It's corpus albicans. Corpus albicans is an actual scar in the ovary. If you look at the ovaries of a 40-year-old female, you will see that they're very scarry. It's very difficult for ovulation to happen when you reach 40 years old because you have been ovulating every month since you were like 12, okay? So you can imagine that every time you ovulate, it leaves a little tiny scar on your ovary. So corpus albicans is a dead corpus luteum and it is an actual scar. What was the function of the corpus luteum?
secrete progesterone and estrogen.
So what's going to happen to progesterone once the corpus luteum dies?
Decrease.
It
Decreases.
drops. Do you see? It starts to drop dramatically. What drop in the progesterone causes? It causes menstruation. You menstruate every time progesterone drops. First of all, what is menstruation? Menstruation is peeling off the endometrium, right? So we made the endometrium thick because we were preparing for the embryo. We were hoping that embryo will arrive, but there is no embryo. Do we need this endometrium? No, so let's just peel it off, right? So this is your menstruation right here. So every time progesterone drops, you're going to have a menstruation. Is that clear? Very important question. What was the function of estrogen in relation to endometrium? It was?
To start build the endometrium.
It was making endometrium thick. Progesterone was keeping it thick. How can progesterone keep the endometrium thick? Inside of the endometrium, we have these vessels. These vessels are called spiral arteries because of their shape. These spiral arteries provide nutrition for the endometrium to make sure that it stays thick, right? The problem is that these spiral arteries are progesterone sensitive. They are only open if progesterone is high. The moment progesterone drops, these spiral arteries constrict. If they constrict, endometrium is not receiving blood. If there is no blood, there is no nutrition. It starts to die. It starts to become necrotic. It starts to peel off. The peeling off of endometrium is called menstruation. So do you get that?
This causes the pain in menstruation.
Yeah. Okay, peeling off endometrium is definitely hurtful. Okay, so it hurts. Okay, so yeah, it's definitely causing the pain. So do you get how progesterone and menstruation are related? Yeah. Is this part clear? Okay, very important thing. How many follicles do we choose monthly?
15 to 20.
To 20.
We said that if you aren't the chosen one, you're going to die. Am I right? You're going to undergo apoptosis and that's it. Are you going to evaporate into the thin air if you die? Of course not. You have to leave a mark. This mark is called corpus fibrosum, or Professor Antal calls it corpus atreticum. So corpus fibrosum or corpus atreticum, these are the follicles that didn't reach the tertiary stage. So if we chose 15, one of them will become eventually after ovulation corpus luteum, but 14 of them are going to be the dead ones, right? So the ones that didn't reach the tertiary stage. So how do we call the 14 of them? We call them corpus fibrosum or corpus atreticum. What is atresia? Atresia means death in Latin. Atresia, atreticum, it means dead in Latin language. Is it clear? Okay, let's imagine I do have an embryo. So somewhere during this time, I had an unprotected sexual intercourse, so I got pregnant. I have an embryo. So, do you think LH, okay, what does embryo need to be alive?
Umbilical cord.
Before umbilical cord. We're talking about first couple of days.
To have
Nutrition?
endometrium thick.
Endometrium.
It needs endometrium because it needs to go here. It needs to go specifically into this thick endometrium and fully be surrounded by it, fully be surrounded by the maternal blood vessels so it can get nutrition from there. So for embryo, it is very important that endometrium is thick, that it stays thick. What can keep endometrium thick?
Progesterone.
So it is in the embryo's interest to keep progesterone stable, to make sure that it doesn't drop, because we have established the moment progesterone drops, spiral arteries constrict, endometrium dies, embryo will go with it, right? What keeps progesterone stable?
The corpus luteum.
What
LH.
corpus luteum
LH.
needs to be alive? It needs LH. So what keeps progesterone stable is LH. So a question, can embryo make LH high always? Can embryo keep LH high? And I really love it because always the boys, they're always like, yeah, and the girls are like, no. Okay, because think about it. What secretes LH?
Corpus, DG.
The pituitary gland.
Pituitary gland secretes LH. Who tells pituitary gland to secrete LH?
GFRH.
GnRH. Who makes GnRH? Hypothalamus. Do you think embryo can control mom's hypothalamus? Okay, so that's why girls are like, I hope not. Okay, I hope this little parasite, okay, is not capable of actually, you know, controlling my hypothalamus. So the answer is no. Even though there is an embryo, even though there is a fertilization, either way, LH will drop. It will always drop. Pregnant or not, LH will eventually come down to its normal concentration. It doesn't matter if you're pregnant or not. So embryo needs to secrete something that looks like LH to fool the corpus luteum. So corpus luteum is going to think it is under the influence of LH and keep on secreting progesterone. How do we call this something that embryo makes that looks like LH? We call it human chorionic gonadotropin, HCG. Anybody knows anything about this one? When I pee on the stick, my urine has HCG. That's the one that we detect on the stick when I pee on it to check if I'm pregnant. Okay, so you check for the HCG. HCG is a substance secreted by embryo. Take a look, it is gonadotropin, so it looks like LH FSH. Your corpus luteum couldn't care less. Pepsi, Cola, same stuff, okay? LH, HCG, they're gonadotropins, it looks the same. So corpus luteum is under the influence of HCG, which I repeat is made by embryo. It keeps on secreting progesterone. Progesterone is stable, embryo stays. HCG is the highest in the first 3 months of pregnancy. But then it decreases because after 3 months of pregnancy, corpus luteum will die. Actually, this transition from first trimester to second trimester, when we're transitioning from the 3rd month to 4th month, this is the most dangerous time for embryo because that's when the corpus luteum dies. But if corpus luteum dies, who secretes progesterone?
Placenta.
Very good, placenta. By the 3rd, 4th time, by the 3rd, 4th month, we already have placenta, and this placenta is busy secreting progesterone. So we don't need corpus luteum, we don't need HCG, we don't need any of this stuff. Placenta will do it by itself. And it's actually quite interesting because placenta takes maternal testosterone and converts it to progesterone. So it takes the most useless thing and converts it to progesterone. So I think it's very interesting. Okay, guys, is it clear? Okay. Let's move on. Fertilization. You know that this is the fusion of the male and the female gametes. Okay, so only 1% of the spermatozoa are going to actually enter the cervix. The cervix is the neck of the uterus. And you remember that spermatozoa can actually survive in the female reproductive tract for 3 to 5 days. The movement from the cervix to the fallopian tube can differ. Some spermatozoa are super, super fast, so it takes them 30 minutes, half an hour, and they're already at the fallopian tube. So can you imagine how fast they swim, like super fast? Some are a bit slower, so it takes them 6 days to reach the fallopian tube. So that's super, super slow. Where does fertilization happen? It happens in the ampullary region. So if I show you, this is the ovary, this is the fallopian tube, this is the uterus. This area right here, this is the ampullary region of the uterine tube. We're going to learn more about it next semester. This is the place where the fertilization will be happening. Okay, why in the ampullary region? Because this is the widest part of the fallopian tube. So supposedly it has the most space. So during ovulation, oocyte is just going here and it stops. It waits for spermatozoas to come here. So advice for girls, you don't approach boys too much. You do one step and they do all this. Okay, so they do all this because this is at the genetical level. This is at the biological level. As an oocyte, as you can see, it's not running towards spermatozoa. It did just 2 steps. It's just in the fallopian tube and it's just waiting for spermatozoas to reach that area. Spermatozoas do majority of the work, right? So from the vagina to the uterus, to the fallopian tube, all the way here. This is a huge, huge way to reach. Fertilization is actually 2 reactions. It's capacitation and acrosome reaction. Capacitation typically happens in the fallopian tube. Okay, so already in the uterine tube, capacitation happens. What is capacitation? This is when we are removing glycoprotein coat from the acrosomal region of spermatozoa. You remember, head of spermatozoa, 3/4 of it are actually covered by acrosome. This acrosome has glycoprotein coat, and what are we doing? We're removing this glycoprotein coat. Why? So we can expose the enzymes. So we can expose these particular digestive enzymes that are needed to penetrate through the zona pellucida, right? Only capacitated spermatozoas are passing through corona radiata. You remember how oocyte looks like now, right? It is a secondary oocyte surrounded by zona pellucida, surrounded by corona radiata. Corona radiata are granulosa cells, right? The ones which have stayed with the oocyte after ovulation. So only the sperms which do not have glycoprotein coat on their acrosome can actually pass through corona radiata and bind to zona pellucida. Once you exposed your digestive enzymes, once you have bound to the secondary oocyte, you're going to see that acrosome reaction begins. Basically, these digestive enzymes start to do their job. They start to actively digest a little piece of zona pellucida, so you can actually kind of sneak into the oocyte this way. What's important is that these enzymes are actually inactive. Oocyte itself activates the digestive enzyme. Within the zona pellucida, we have a protein which is called ZP3. And this zona pellucida protein 3 is the one which activates the digestive enzymes of the acrosome of spermatozoa. Is that clear? Basically, the enzymes are inactive. So only when they bind to zona pellucida will activate the enzymes back, right? So it will make sure that they can actually do their job properly. Once zona pellucida is digested, I think it's important to mention that it's not the whole thing which is digested, just a little bit of it, just a little tiny window. Only the head of spermatozoa squeezes into the cytoplasm of the oocyte. The head contains nucleus. The nucleus of the spermatozoa will fuse with the nucleus of the oocyte. So I repeat, I draw it for you here weirdly. It's not the whole spermatozoa that goes inside, just the head. So neck, middle piece, principal piece, and the tail, they are left behind, okay? They are not entering the oocyte, only the head. What are the main results of fertilization? This is a question in the exam. First of all, we restore the diploid number of the chromosomes, because when the two nuclei fuse, when the nucleus of the spermatozoa fuses with the nucleus of the oocyte, we now have a fertilized oocyte. Oocyte, as you remember, always carries X. Spermatozoa could carry X or Y. So when they fertilize, you already know if you have a girl or a boy. At the genetic level, this one cell already has XX or XY. It's just that we do not have the technology to check it. We do not know, right? How do we call the fertilized oocyte?
Zygote.
Zygote. Yeah, so this is known as the zygote. So you determine the sex of the zygote at the genetic level, and you initiate the cleavage. Okay, something is more important here. We said digestive enzymes. What are their names? Acrosin and trypsin-like. That's the enzymes. So acrosin and trypsin-like enzymes, they need activation by zona pellucida, right? So they need activation by ZP3. Here's some additional information, but it's not important, okay? So maybe the most important is that you deposit 200 to 300 million of spermatozoa. How many of them are capacitated? I hope you remember, right? So capacitation is where exposing the digestive enzymes, only 500 of them. So millions are deposited, only 500 are capacitated. So only 500 actually have the ability to fertilize the oocyte. Once zygote is present, once zygote is made, zona pellucida changes its composition. It's going to undergo cortical and zona reactions. You're going to see that the zona pellucida starts to really stiffen. It starts to become harder. There's some kind of conformational change in the proteins here. As a result, it becomes harder. Why? To prevent polyspermy. We do not want our spermatozoa to get in. We already got our spermatozoa, right? So we already have the zygote, so everything is great. So we don't need it. So you're going to see that definitely this happens, that zona pellucida changes itself. It changes, it has some kind of conformational reaction. And we said we start the cleavage. So what is cleavage? Cleavage is division. We start to divide. So in the ampulla, we now have a zygote, one cell, fertilized oocyte. Inside of the zygote, in the nucleus, we either have XX or XY, right? Boy or a girl, one of the two. What is the zygote surrounded by? It is surrounded by zona pellucida. And the zona pellucida is very, very thick. It's not your typical one. It's extra thick, extra tough, right? One cell divides to become two cells. Now I have a structure that contains their two cells. Two cells become how many?
4,
4 become
8,
8. 8 become
16.
16. These stages are important. These are the cleavage stages of the zygote. Typically, cleavage takes 2 to 3 days from the moment of fertilization. And the names of the structures are the following. You have zygote. Zygote becomes 2-cell stage. 2-cell stage becomes 4 and 8. Together they have a name. They're called blastomere. So a structure that possesses 4 cells, this is called blastomere. And 8, it is also known as the blastomere. And the structure that possesses 16 cells is known as the morula. Morula is the one which is at the border. Do you see? It is the one which is about to enter into the uterus. Why do we call it morula? Because it resembles mulberry. It looks like a berry. Okay, it looks like a berry. It has these 16 cusps. A very important question. How is the size of the zygote and morula? Is it different or is it the same?
Same.
Size of the zygote and morula is going to be the same. They're identical in size. What is the only thing which is different?
More cells inside.
Number of cells. Why morula is not increasing in size?
Maybe because it doesn't
The
have any
tube.
space in the fallopian tube.
Not that it doesn't have space in the fallopian tube, but rather what surrounds it. What is outside of the structures?
Collagen.
Zona pellucida. Don't forget that here we have a very thick zona pellucida, and it doesn't allow the cells to expand. It keeps them intact, okay? It keeps them fully closed up. So the reason that morula does not expand is because it has no space to expand. The space is limited by zona pellucida. Is this clear? So this is the third day after fertilization. You're gonna see that morula is the one that enters into the uterus. In the uterus, morula is going to become another structure, which is called blastocyst. So how does it happen? How does morula become blastocyst? I think it's important to mention, until morula, so including morula, these cells are stem cells. Which kind of stem cells?
Pluripotent.
Totipotent. Yes, they're totipotent. Very good. So zygote is a totipotent stem cell. Two cells, totipotent. Blastomere, toti. Morula, toti. They can become anything. Anything. So whenever we, hypothetically, we could grow organs, right? Hypothetically. All we have to do is take this. We just have to take this morula, force it to become a certain human being, and then harvest the organ. That's it. Is it ethical? Not really. Okay, so that's why we don't do it. Okay, so morula, until morula are totipotent, remember that. So morula enters into the uterus. Inside of the uterus, we have lots and lots of fluid. 60% of you is water. It is normal for droplets of fluid to accumulate in your body somewhere. Okay, so it is very normal. So we have a little bit of droplets of fluid here, a bit here, a bit there. Due to osmosis, when morula enters into the uterus, this fluid wants to enter into morula, because from higher concentration, which is the uterus, it wants to go to the lower concentration, which is the morula. Is that clear? So now I have here morula, 16 totipotential stem cells surrounded by zona pellucida. Now the fluid tries to go into the morula. Can it? Not really, because there is zona pellucida. But does it give up? No. So slowly it starts to dig into the morula. And as it tries to enter the morula, it slowly washes away zona pellucida. It just washes it away. So now your morula grows, right? Every little step it makes, every little millimeter it takes, it divides. And as it divides, it becomes bigger and bigger and bigger, because there is no zona pellucida, because fluid is entering into your morula. When fluid enters into morula, it doesn't just accumulate randomly. It's not like there's a droplet of fluid here, droplet of fluid there. It's not like that. Morula kind of separates the cells to make sure that it has a cavity for the fluid. You're going to see that some cells of the morula start to accumulate in the center, and some cells start to accumulate in the periphery, these cells. Why? To make sure that there is a cavity, so fluid can actually enter into that cavity. This structure is definitely not morula. This is called blastocyst. What does blastocyst consist of? You can see that the cells that are staying in the center, this is known as the inner cell mass. The cells which are staying in the periphery, they are known as the outer cell mass. And the cavity with all that fluid, this is just your blastocyst cavity. Another name for the inner cell mass, it is also known as the embryoblast. Another name for the outer cell mass, it is also known as the trophoblast. But Professor Antal calls embryoblast embryo proper. So I repeat, this huge thing is a blastocyst. Could you tell me, where did you come from? You, where did you come from? From which part of a blastocyst? From a whole thing or from some little thing?
Inner cell mass.
Inner cell mass. As the whole human arises from here. So only embryoblast, only embryo proper gives rise to the entire human being. What does trophoblast gives rise to? Gives rise to placenta. Trophoblast is the future placenta. Embryoblast is the future human being. So a very important question. Which stem cells are trophoblast?
Plural.
Mesenchymal stem cells.
Mesenchymal stem cells are pluri as well. So they aren't pluri.
Tati.
Aren't toti.
Multi.
They're multi. Why trophoblast is a multi-potential stem cell?
They can only become one thing.
They can only become one thing. And what is that one thing?
Placenta.
Placenta. So trophoblasts are multi-potential stem cells because they can only become placenta. What about embryoblast? Which kind of stem cells are they?
Pluripotent.
Pluri. Why not toti?
They cannot become everything.
So what exactly can it not become?
Placenta.
Placenta. Embryoblast cannot become placenta, so they can't be toti. They have to be pluri. Okay, embryoblast is a pluripotential stem cell. Is it clear? Is there zona pellucida outside of the blastocyst?
No.
Nope. The next step is implantation. So now your blastocyst is in the uterus, it is ready to implant. Implantation starts around day 6. Okay, so after fertilization. So this is what you can see here. This is your endometrium. It's thick and nice, ready for implantation. This is your blastocyst, ready to be implanted in it. So how is it happening? It happens due to our very important structures, which are called integrins and laminins. Integrins are located on the surface of the trophoblast. Laminins are located on the surface of endometrium. So here we're going to have lots and lots of laminin. This laminin is ready to interact with integrin. Where is integrin? We said trophoblast, but not everywhere on the trophoblast. Integrin is present only on that trophoblast which is overlying the embryoblast. Only that trophoblast, which is on the top of the embryoblast, will have integrin on their surface. So what is going to happen? Laminin attaches to integrin, your blastocyst is attached to the endometrium. Is it implanted though? No. Okay, so think about boobs implant or ass implants or whatever. They have to be fully inside. It's not like there's just a part of it hanging outside. Okay, so not really. They have to be fully inside of whatever you implant into. So this structure is not even close to being implanted. So initial attachment, initial adhesion is definitely because of the laminin and integrin. But what actually is the driving force for implantation? This is a histo question. Integrin loves who?
Fibronectin.
Amazing. So who must have fibronectin?
Endometrium.
Yes, endometrium is filled with fibronectin. The moment integrin is aware of that fact, nothing will stop it. It will start to invade the entire endometrium to be fully surrounded by the fibronectin. This is why some girls will bleed. Okay, some women will bleed and they will mistake it for menstruation. But in reality, it's not a menstruation. It's an implantation bleed. But a smart girl will realize that this is not menstruation, because you wouldn't bleed for like 3-4 days like you normally bleed. It will be just a bit of blood, okay? But it's not like your proper menstruation. So you get it? Initial adhesion, laminin on the endometrium, integrin on the trophoblast. But the actual implantation is integrin fibronectin interaction. Integrin won't stop until it is fully surrounded by the fibronectin. So it has to be in the endometrium. There are some places where we want our embryo to be implanted. This is going to be anterior, posterior side of the uterus between two uterine glands. Uterine glands are epithelial glands which are present in the endometrium. They're just like your sweat or salivary glands, but they're sitting in the endometrium. Uterine glands are very rich in glycogen. And you remember that glycogen is the polymer of glucose. So do you get why we have to be implanted there? God knows when nutrition is coming in. So definitely I want to be implanted in this area, right? How do we call the pregnancy which is outside of the uterus? For sure you know. We call it ectopic. Have you heard this word before? Ectopic pregnancy. Ectopic pregnancy is quite common, especially nowadays with our generation, mine for sure, I think yours as well. Smoking, drinking, stress. Our parents didn't do a good job raising us as well because they were busy, okay? So we ate sugar, I don't know, pollution, all of this stuff. So definitely very big thank you for that. But yeah, okay, so ectopic pregnancy is definitely a big thing. And it is a pregnancy outside of the uterus. Most common, it's in the fallopian tube. So basically your zygote doesn't move. It stays there. It just stays in the fallopian tube. It doesn't move. What do you think moves the zygote in the fallopian tube to the uterus?
Water.
Prostaglandin.
Cilia.
Cilia, very good. This is a histo thing, cilia, motile cilia. There's lots and lots of cilia that are sitting here, and they're swiping, they're rolling your zygote towards the uterus. Guess what kills cilia? Untreated chlamydia. How often is chlamydia nowadays? Super freaking often, okay? University of Debrecen, guys, fun fact, I was working a little bit in the dermatology department, okay? It's crazy. If I can give you any advice, do not have sex in Debrecen, please, because this is insane, okay? So this is a filled STDs kind of pothole, okay? So do not do that, because it's very, very common, especially in Hungary as it is. The only thing that is not common in Hungary is HPV. Okay, it's common everywhere else, but not in Hungary. It's the one that causes cervical cancer. Yeah, so the one that causes cancer of the cervix in female. But it's not common because Hungary actually vaccinates girls and boys against this HPV, which is a very good thing. But everything else is super common. So untreated chlamydia, keyword untreated. If you had it, you got yourself treated, you're okay. But untreated, you're unaware that you have chlamydia, can definitely kill these cilia right here. That will lead to infertility because they can't swipe the zygote to the uterus. You have to do artificial insemination or something like that, right? So, or just artificial implantation. So you have to do it like in a hospital to implant the embryos right here. So this is the most common one. The other common place for ectopic pregnancy is the ovary. So maybe it didn't rupture enough. Okay, maybe there wasn't enough of a force. So it ruptured a little bit, but the oocyte never left the ovary. For spermatozoas, it doesn't matter. They're just gonna go this way. Okay, so just extra, I don't know, mile, come to the ovary, fertilize the oocyte, that's it. You have an embryo in the ovary. And the least uncommon is pouch of Douglas. I can't tell you more, okay? Because I don't want to go into the whole anatomy of how. We will learn it next semester, anatomy how. For now, just know that pouch of Douglas is the third place where ectopic pregnancy could be happening. Pouch of Douglas is the space between uterus and rectum, okay? It's the deepest point in the female's body, so it can definitely be there. Next class, I will tell you a fun story that happened in Hungary, which is crazy, okay? But not right now because we're running out of time, okay? Because the baby survived here in Hungary, 9 months. It's insane. Like, it's not possible, but it happened, okay, in Hungary. But yeah. Okay, we talked about this.
Sorry, but how does the embryo get its nutrition in ectopic pregnancy?
Next class, we will for sure talk about it. It can't. Ectopic pregnancy is not survivable. If you have ectopic pregnancy, the embryo will die. It will rupture these areas. The mom will die from bleeding if it's not caught early. Okay, so ectopic pregnancy is a very, very dangerous thing because it can lead to so many things, to shock in mom, sepsis, etc. It can lead to death in mom. Ectopic pregnancy is not viable. You can't survive as a baby from ectopic pregnancy unless you're a gypsy in Hungary, because then you can. Okay, actually happened. Okay, about 3 years ago, a baby survived right here. It was a pregnancy in the pouch of Douglas. How the hell did it survive? Nobody knows. But I think it's a great story to tell that baby, like ectopic, not even in the uterus, yeah, ectopic. Next class, I'll for sure tell you the story. Okay, guys, we talked about this. You remember all of the different stages. You remember this part, right? So what's happening to the endometrium, spiral arteries, we talked about it. Now we are reaching this part, and these pictures are crucial, guys. Please spend some time on these pictures. They will bring these pictures for sure. So what do you see? This is your blastocyst, and you can see that it is implanted, more or less. It's not fully implanted because the ass of it is still hanging outside of the endometrium, but still, it's starting its normal implantation. This is day 8. This is endometrium. You can see that these are uterine glands. Uterine glands, as you remember, are rich in glycogen. So this is a very good source of nutrition for my blastocyst. During day 8, you're going to see that blastocyst starts to differentiate. Before we talk about it, could you quickly tell me, how do we draw a blastocyst? What does it have here?
Embryo proper.
So
Fluid.
here, this will be the embryo proper. This is the inner cell mass or the embryo proper. What about this?
Cavity.
This is the cavity. This is the
Trophoblast.
cavity. Yes, this is the trophoblast. This is the outer cell mass or the trophoblast, right? So what is this?
Trophoblast.
Yes, this is the outer cell mass or the trophoblast. What about this?
Embryo proper.
Embryo proper or inner cell mass. What about this?
Cavity.
Cavity. Okay, so this is going to be the cavity. You're going to see that on day 8, your outer cell mass differentiates into 2. You see that there is the outer trophoblast, which is known as the syncytial trophoblast. And there's also inner trophoblast, which is known as the cytotrophoblast. So the outer cell mass differentiates into 2. Okay, it differentiates into 2. The outer syncytial, the inner cytotrophoblast. Syncytial trophoblast is the one which is multinucleated. It has lots and lots and lots of nuclei, and it has no cell borders. It's just a jelly that consists of syncytial trophoblastic cells. Okay, just a jelly. Syncytial trophoblast is the one which secretes HCG. You remember? Human chorionic gonadotropin. So HCG secretion begins on day 8. So is it useful to do a pregnancy test the day after you had unprotected sex?
No.
Absolutely not. You need to wait lots of days. Actually, the longer you wait, the more accurate the result will be. On average, you wait 2 weeks. Or the day after the sex, you just do plan B, right? The pill, and it drops your progesterone. If you drop the progesterone, what's going to happen? You'll menstruate. There is no endometrium. So even if you do have an embryo, it has nowhere to implant. So you will have that. So remember that this is my HCG, and it is done by syncytial trophoblast. Its production begins on day 8. Cytotrophoblast that you see here has 1 nucleus per cell, and it has borders. Do you see? It does have the cell membranes. So again, as an exam, they will ask you this question: what is the differentiation of the trophoblast? Differentiation of the trophoblast is that it divides to become syncytial and cyto. When? On day 8. Simultaneously, on day 8, your inner cell mass also divides into 2. Into the top layer, which consists of tall columnar cells that are known as the epiblast, and into the bottom layer, which consists of the small cuboidal cells called hypoblast. So inner cell mass differentiates into 2 as well, the epi and the hypoblast. Together, epi and hypoblast are known as the bilaminar germ disk. So a very important question: when does bilaminar germ disk appear?
Okay.
Day 8. Out of what?
Inner cell mass.
Inner cell mass. It arises out of the inner cell mass. It arises out of the embryoblast. On day 8, simultaneously, the cells of the epiblast form a cavity, which is known as the amniotic cavity. So inside of them, somewhere there is an amniotic cavity right here. All the additional information is here. Just read it. We're moving on. So this was day 8. Now we're looking at day 9. Is your blastocyst implanted? Yes, thanks to this structure, thanks to the fibrin coagulum. You can see that fibrin coagulum is like a door that shuts the entrance point of the blastocyst, right? So it makes it fully implanted in this area. How do we call this?
Germ disk?
Bilaminar germ disk. Bilaminar germ disk consists of the top
Epiblast.
and bottom
Hypoblast.
hypoblast. How do we call a cavity within the epiblast?
Amniotic.
Amniotic cavity.
Yes. So how do we call this one? We just call it blastocyst cavity, because last time we checked, this was just a blastocyst cavity, this one. How do we call the outer layer? Syncytial trophoblast. As you remember, it consists of the multinucleated cells without cell membrane. It's just a jelly that consists of some kind of cells without the plasma membrane. The inner is, of course, the cytotrophoblast cells. Now, there is a new structure which is known as the exocoelomic membrane or Hughes membrane. Can never pronounce this name, okay? Everybody says something else. Exocoelomic, exocoelomic, I don't know. Just call it Hughes membrane, okay? So this is the Hughes membrane. Hughes membrane consists of the cells of the hypoblast. You're gonna see that cells of the hypoblast starting on day 8, they're starting to detach and literally line the wall of the blastocyst cavity. Okay, so they keep on lining the wall of the blastocyst cavity right here. Now, the moment Hughes membrane is present, the name of the cavity changes. It becomes primitive yolk sac, or another name for it, exocoelomic cavity. So could you tell me what is the only difference between blastocyst cavity and primitive yolk sac? What is the only difference?
Membrane.
Hughes membrane, presence of the Hughes membrane. That's the only difference between the two cavities. Okay, that's the only difference between them. They're basically the same cavity, right? So just add the Hughes membrane, and this is your primitive yolk sac. On day 9, you're also going to see that syncytial trophoblast has some kind of holes. We're medical students, we can't say holes. We replace it with a Latin name. We call it lacuna. Okay, so syncytial trophoblast starts to possess some kind of spaces, some kind of lacunas. These trophoblastic lacunas are very important because this is the area where maternal blood vessels are going to be entering. Okay, so this is the part where maternal blood vessels are entering. Okay. Next day is day 11, 12, right? So this is day 9. So outer layer.
Syncytial trophoblast.
Trophoblast, inner layer. Cytotrophoblast.
Bilaminar germ disc. Amniotic cavity. Primitive yolk sac.
Primitive yolk sac. What is supposed to be here?
Hughes membrane.
Very good. It's this yellow tissue, right? So the hypoblastic Hughes membrane. On day 11, 12, you're going to see that Hughes membrane starts to split. Spaces are appearing within the Hughes membrane, and they're dividing your one Hughes membrane into two. So do you see there is a space appearing, and as a result, it divides your one Hughes membrane into two. These are the longest names in embryo. Nothing is longer than that. So that part of the membrane, which is closer to the cytotrophoblast, that part of the Hughes membrane is known as the extraembryonic somatopleuric mesoderm. Extraembryonic somatopleuric mesoderm is this part, the one which is closer to the cyto. These are these spaces, right? So the ones which divide your Hughes membrane, these are the spaces. So the one part of the Hughes membrane which is closer to the yolk sac, this is known as the extraembryonic splanchnopleuric mesoderm. What are the two words that are in both of them? Extraembryonic mesoderm, right? Extraembryonic mesoderm. So where does extraembryonic mesoderm come from?
Hypoblast.
You're right, just don't forget, it comes from
Bilaminar germ disc.
It comes from
Hughes membrane.
Hughes membrane, and Hughes membrane comes from hypoblast. So technically, you're right to say that it comes from hypoblast. Yeah, you're right to say. Just it's a bit incorrect. Okay, so you have to say that it comes from the Hughes membrane, which in return comes from the hypoblast. Is that clear? Why do we call it extra embryonic? Where is the embryo on this picture?
It's the disc, the germline disc.
It's the bilaminar germ disc. Okay, so this disc right here, the bilaminar germ disc, that's your embryo. That's the human being right there. Everything else is extra. So as a result, this tissue is the extra embryonic mesoderm. I told you that blood vessels are going to be entering into the trophoblastic lacuna. You remember that these trophoblastic lacuna were spaces, right, within the syncytial trophoblast. When the blood vessels enter here, you're going to see that we establish uteroplacental circulation. So when does uteroplacental circulation is established? On day 11, 12. What does it mean? it means that blood of the mom starts to flow here, okay? And it brings the nutrients. We're starting to make placenta, okay? On day 11, 12, we're slowly starting to make placenta. Is it clear? Okay, last thing for these slides. These spaces, which are within the Hürsos membrane, between somato and splanchnopleurotic mesoderm, these spaces want to become confluent. These spaces want to fuse. They want to become one huge cavity. And this is what you can see on this picture. Do you see this huge cavity right here? This huge cavity is a product of the fusion of these spaces right here. So when these little tiny spaces have fused together, you have made this one huge cavity. How do we call this one huge cavity? We call it chorionic cavity. Okay, we're gonna call it chorionic cavity. Another name for it, extra embryonic cavity. So could you tell me the layers? What is the outer layer?
The
Syncytiotrophoblasts. This?
Cytotrophoblast.
Cytotrophoblast. What is this?
The H membrane.
Yes, but which part of the Hürsos membrane?
Somatic mesoderm.
One. Okay, so it's this part right here. So this is the extra embryonic somatopleuric mesoderm. Together, syncytio, cyto, and somato, together, they're called chorionic plate. It's a mistake. Okay, so together, they're known as the chorionic plate. Next thing we have is the chorionic cavity. It's this little tiny spaces here that have fused together. The next thing we have is the yolk sac. Don't forget what lines the yolk sac. What lines the yolk sac from here to here?
This membrane.
Very good. The splanchnopleurotic mesoderm. Okay, you're going to see that the splanchnopleurotic mesoderm is the one that starts to line this tissue. Now, what is this?
The germ disc.
Bilaminar germ disc. This is amniotic cavity, right? And now the big question. What is here? What is on the top of the amniotic cavity, this pink tissue? I'll help you. Is it somato or is it splanchno? Somato or splanchno?
Somatic.
I hear the two. Let's follow. This is where the splanchno starts. It goes around the yolk sac. It finishes around the yolk sac. It finishes here. This is the amniotic cavity right here. This is cytotrophoblast. Which Hürsos membrane, which part of the Hürsos membrane is going to go this way? It's going to proliferate this way, this somato. Okay, so the amniotic cavity will be covered by the somatopleuric mesoderm. Okay, by the extra embryonic somatopleuric mesoderm. So this is very important. We have 3 cavities. Which extra embryonic mesoderm will be covering yolk sac? The extra embryonic splanchnopleurotic. Which extra embryonic mesoderm will be covering the amniotic cavity?
Somato.
Somato. Which one lines the chorionic cavity?
Somato.
Also somato. So for the self-control test, remember that somato lines 2 cavities. It lines the chorionic cavity and it lines the amniotic cavity as well. Yeah. There is a structure here which is called connecting stalk.
Excuse me?
Connecting stalk is a substance where we have both, where we have the somato and splanchno pleuric mesoderm. Connecting stalk is your future umbilical cord. I'm a true Russian. I hate sun. I'm so close to the Siberia where we don't have sun whatsoever. Every time the sun arises, I hate it. Yes, okay, so you're going to see that the connecting stalk is the future umbilical cord, and connecting stalk is going to be consisting of two mesoderms together. It consists of the somatopleuric and splanchno. It's like a meeting point for both of them. Okay, last thing here. It says secondary yolk sac, because last time we checked it was primitive. So what happened? By the way, this is day 14. I'm not sure if I wrote it here. I didn't. Okay, so this is day 14. This is second week. Why is it the secondary yolk sac? It is the secondary yolk sac because the original one has bursted. Can you imagine when you were forming the chorionic cavity here, the amount of pressure that was exerting on the original yolk sac is insane, right? So you're constantly bugging the original yolk sac non-stop. As a result, it bursts. It can't withhold much pressure, so it bursts. The problem is that the yolk sac is a very important embryonic organ. Yolk sac is the first hematopoietic organ. It's the first organ to make blood. So you bursting this yolk sac by trying to form chorionic cavity is a fatal mistake. You just bursted the first organ to give blood to the embryo. Yeah. So what do we need to do? We need to repair it. How can I repair it? I'm going to send cells of the hypoblast. You're going to see that some cells of the hypoblast detach. They start to migrate around the wall of the yolk sac and patch it up. They just make additional layer in the yolk sac. So they patch it up a little bit, yeah? So from day 14, you will not only find cells of the hypoblast in the bilaminar germ disc, you will also find cells of the hypoblast in the wall of the yolk sac as well. From day 14, hypoblast is not only in the disc, it's also in the wall of the yolk sac. Is that clear? This patched up yolk sac is called secondary yolk sac, and it is much smaller than the original one. Original one was this big. This is why sometimes you see a remnant of the original yolk sac in the chorionic cavity. That remnant is known as the exocoelomic cyst. And on this picture, they're trying to show you how it is dripping down. Do you see that this cyst is literally kind of dripping down this way because this one is leaking. It's patchy, right? So it needs additional cells of the hypoblast to repair that area. So I repeat, exocoelomic cyst is just a remnant of the original yolk sac. And it is in the chorionic cavity. Do we have any questions here?
Can you please repeat what lines the yolk sac again?
Absolutely. So I go back here. We have Hürsa's membrane. Hürsa's membrane comes from the hypoblast. The problem is that within this Hürsa's membrane, spaces start to appear. These spaces divide your one Hürsa's membrane into two. The part which is lining the yolk sac is known as the extraembryonic splanchnopleuриc mesoderm. The part of the Hürsa's membrane which is lining the cytotrophoblast, so this area, is known as the extraembryonic somatopleuric mesoderm. The spaces between these two mesoderms are going to fuse and form a cavity. How do we call this cavity? We call it chorionic cavity. So outside of the yolk sac, I still have splanchno. Do you see? I still have splanchno. Outside of the chorionic cavity, I still have the somato. The big question is, what's up with the amniotic? Who is going to be covering the amniotic? Because cytotrophoblast is on the top of the amniotic, it must be somato, right? Because somato is the one which will be lining the cytotrophoblast. So it lines the amniotic as well.
Okay, thank you.
So that's...
Sorry, I have a question.
Yeah.
So what's the difference between the hypoblast cells in the Hürsa's membrane and the ones that line the secondary yolk sac?
Very good question. without going too much details, the cells in the Hürsa's membrane are given rise to mesoderm. Mesoderm is different from hypoblast. Think about it this way. Hypoblast is like a stem cell. Mesoderm is like a differentiated cell. Okay, so the main difference between Hürsa's membrane and hypoblast is that in the Hürsa's membranes, the cells are adults, right? So they're already differentiating to become something already.
Okay,
While the hypoblast is still like a baby.
thank you.
Welcome. Okay, so I'm going to let you go. We're missing like 20 minutes, even 22 minutes, but I'm going to add them next class. I am fried. I'm sorry, guys. I've been tutoring nonstop since 8 a.m.